Mucor. Mucor is one type of fungi. It developed on the bread. It's similar to the rhizopus. So these are also called bread molds or pin molds. Pin molds means during the asexual reproduction, they can produce sporangiums as a pin-like structure on the mycelium. The sporangiums are blue in color sometimes, they are green in color sometimes, they are black in color sometimes. Based on the color of sporangium, they called blue fungi, green fungi, or black fungi. Now we discussed about the Mucor classification. Here it is the classification of Mucor, division Eumycota, subdivision Zygomycotina, order Mucorales, family Mucoraceae. Now we discussed about the mycelium structure of Mucor. It is also similar to the filamentous branched unseptate like structure. The cell wall of the mycelium containing cellulose, hemicellulose, and chitin and monon proteins also. It gives a perfect shape to the mycelium. It protected to inner parts. Below the cell wall, a thin plasma membrane is present. The plasma membrane mainly made with phospholipids and protein chemicals. Below the plasma membrane, protoplasm is present. In that protoplasm, more number of haploid nuclei are present. Around these nuclear envelope is present. The nuclear envelope contains nuclear pores. Below the nuclear envelope, nucleic acids are present. Here in this mycelium of Mucor, more number of haploid nuclei are present in the filamentous. This position is called senocytic position or multinucleate position. And around these nucleus, endoplasmic reticulums are present. The endoplasmic reticulums covered with ribosomes. If ribosomes are present in endoplasmic reticulum, they called rough endoplasmic reticulum. The rough endoplasmic reticulum mainly helps in the synthesis of protein. And near these endoplasmic reticulum, mitochondrions are present. And sisters of the mitochondria called mitochondrion, and sisters of the mitochondria called mitochondrion. The mitochondrions mainly helps in production of ATP energy to the mycelium. And near these mitochondrions, vacuoles are present. They act as excretory organs of the mycelium. If any waste materials are present in the mycelium, they are stored in the vacuoles. In this cytoplasm, ribosomes also present. They are mainly helps in the production of proteins. And in this mycelium, reserved food materials also present. The reserved food materials are called oil droplets. They are present on the mycelium of the peripheral regions. And liquid cytoplasm also present. It is the mycelium vegetative structure, Mucor mycelium vegetative structure. Now we discussed about the vegetative reproduction of Mucor. Mainly the vegetative reproduction done by the Mucor by the two methods. One is chlamydospores and idiospores. Chlamydospores are developed in the unfavorable conditions. During the unfavorable conditions of the mycelium, the nucleus, around the nucleus, thick cell wall is formed. Below the cell wall, nucleus with cytoplasm is present, called protoplasm. The chlamydospores are in rectangular structures. At the matured stage, the chlamydospores are leave from the mycelium. They are developed into germinative. If they are reached comfortable in place, the germinative develop into new mycelium. This reproduction is also called vegetative reproduction. Sometimes we call it asexual reproduction. Now we discussed about the asexual reproduction. Asexual reproduction of mycelium done by the favorable conditions of the Mucor. During the favorable conditions of the mycelium, aerial stalk-like structures are developed. The aerial stalk-like structures are called sporangiopore. On the terminal part of sporangiospore, more number of haploid nuclei are reached. They are develop into a dome-shaped or club-shaped structure called sporangium. The sporangium containing more number of nuclei of haploid nuclei and cytoplasm. This sporangium part is separated from the remaining mycelium by the columella. Here it is in this columella, more number of vacuoles are present. The vacuoles are present at the terminal part of sporangiospore. The part is called columella. When the matured stage of the sporangium, all haploid nuclei are converted into haploid spores. When the matured stage, the sporangium wall is broken down and haploid spores are comes out from the mycelium or from the sporangium. They are landing on the mature substance. They are developed into again new mycelium by the producing of germitube-like structure. When the matured stage, we observed a collar-like structures. If they are dehiscensed, the sporangiums are released spores. They observed a collar-like structure at lower part of sporangium. Now, the sporangium produced more number of haploid spores. They are landing on this moisture substance or the sugar content or on the food materials. They produce germ-tube-like structures. They can develop into new mycelium. Now we observed sexual reproduction of the mucor. Mucor participated in the sexual reproduction in unfavorable conditions. Around the mycelium, unfavorable conditions are present. They are participated in the sexual reproduction by the conjugation mechanism. See, here it is the before the unfavorable conditions, the mycelium is unique structure. If unfavorable conditions are developed, the mycelium divided into plus strains fungi and minus strain fungi. Plus and minus strain fungi are produced two different types of sporangiums, like club-shaped structures in the opposite manner. From the somatic hyphae, from the two different somatic hyphae, lateral side in opposite manner of two fungi, they produced a club-shaped structure. The club-shaped structures are containing haploid nuclei on the terminal part of club-shaped structures. The club-shaped structures or complete part is called gametangium. The gametangiums are containing one suspensor. Here it is two strains, two different strains. Plus strains fungal hyphae produced one gametangium. Minus strain fungus produced another gametangium. The two gametangiums are containing suspensors at the back side. The two gametangiums are in contact position. By the contact method between the two gametangiums, canal-like structure is formed. The canal-like structure is called conjugation tube. By the conjugation tube, two different from the two different gametangiums, the different two nuclei are fused to form diploid zygotes in the conjugation tube-like structure. See, here it is, the complete structure is called zygospore. The zygospore participated in the germination. The zygospore is in diploid condition. They develop into sporangiopore. On the sporangiopore, more number of diploid nuclei are present. The diploid nuclei are participated in the meiotic division. If diploid nucleus participated in the meiotic division, they can develop into haploid spores. Haploid spores are moved by the wind and landing on the moisture substance with nutrients. They are developed into germitube. The germitube again develop into somatic hyphae of mucor. It is the continuation life cycle. We observed in the life cycle, here it is only diploid phase present in the zygote, but remaining all stages are in haploid condition when they participated in the vegetative propagation, when they participated in the sexual propagation, sexual reproduction, asexual reproduction. During the asexual reproduction, we observed only diploid nucleus in the zygote. Remaining all stages are in haploid condition. So this type of life cycles are called haploid life cycles. So in mucor, haploid life cycles are present.